All right, let's start off just answering this question. If you said all of these choices are correct, that is the correct choice. So when we talk about the evolutionary success of eukaryotes, there's a couple major traits that we talked about. One is possessing a dynamic cytoskeleton. So dynamic, remember that just means the cytoskeleton can move around, and this allows them to change shapes and do things like phagocytosis. Having complex patterns of gene regulation. So we talked about this idea that they have linear chromosomes, and that allows gene replication to occur, or gene expression to occur at lots of different locations, and also allows other sort of regulating factors to control whether genes turn on or off, or how they're expressed in terms of the degree that they're expressed. And that's important to be able to sort of regulate these different patterns of gene regulation and gene expression so they can have different cell types and turn different genes on and off. And this is really important for the evolution of complex cell types and multicellularity. Talked about concentrating where energy metabolism takes place. So this is really all about having organelles. So things like photosynthesis, things like cellular respiration occur in mitochondria and chloroplasts that concentrate different sort of chemical reagents and allow those different metabolic processes to occur much more efficiently than they would if those organelles didn't exist. And that's really important, once again, for the evolutionary success of eukaryotes and enabling them to produce enough energy to be able to grow to the sort of large sizes that they have. And also possessing a dynamic endomembrane system. So remember that dynamic endomembrane system is basically a system of membranes that can move around and move stuff all around the cell, whether it's waste moving out or energy and different products moving in and around a cell. That's all very important for maintaining the larger size that eukaryotes have, so that those things can move in and out of a cell faster than diffusion alone would allow them to. Next question. If you said all of the above, you are correct. So cell adhesion was really important to enable cells to stick together. Structures such as plasmodesmata and gap junctions. So remember, plants use plasmodesmata. Plant cells use plasmodesmata. Animal cells use gap junctions, basically to allow communication and signaling in between cells. So these are sort of holes and channels that connect individual cells to allow them to communicate with one another, which is really important for multicellularity. Regulation of gene expression within and between cells. So once again, regulating gene expression is basically turning on and off, or up and down, how much genes are expressed. Remember, when we're talking about genes being expressed, we're talking about genes. That's DNA that's coding for RNA. That's RNA that's coding for proteins. Those proteins hold different traits, or can be even signaling molecules that allow cells to communicate with each other and to communicate internally within a cell as well. And all of that regulation of gene expression, both within and between cells, is really important for coordinating interactions between cells, and it's a key part of multicellularity. Lastly, tissues and organs that use bulk flow to transport nutrients, oxygen, and molecular signals throughout the body of a multicellular organism. So diffusion alone is not enough to sort of get nutrients into all the different cells of an organism, especially because in multicellular organisms, those cells aren't exposed directly to the environment. So those individual cells need a way to get the waste out and out into the external environment, and to get nutrients and things that they don't have access to into those individual cells. And bulk flow is a great way to do that. So all of the above. Next question. The evolution from simple multicellular organisms to complex multicellular organisms required which of these things? You got to choose one. Said the evolution of a system that enabled bulk flow. That is the correct answer. So if you remember, there's a sort of stepwise sequence of events that needed to occur in order for multicellular or complex multicellularity to work, in order for complex multicellularity to evolve. And so if we look at things like adhesion and cell signaling, that's something that evolved, and we see that even within single-celled organisms. So these groups called the choanoflagellates, those can be single-cellular things, and they can still do adhesion and cell signaling. But the ability to communicate in between individual cells once they're sort of adhered to one another, and also to have bulk flow so that nutrients can flow into and out of organisms and reach all the cells that make up these multicellular organisms. Those are two key aspects that needed to happen to go from this idea of simple multicellular organisms to complex multicellular organisms. And remember, complex multicellular organisms are those organisms that we normally think of when we think of a multicellular organism that have different specialized cell types than those individual cells that make up a complex multicellular organism cannot survive on their own. They need the other cells, or they rely on the other cell types, other tissues, other organ systems to survive. Okay, true or false? This is a false statement. So remember that diffusion is a passive process. It doesn't require energy, and that molecules and chemicals and anything else are naturally always going to do diffusion. They're always going to diffuse from places of higher concentration to lower concentration just through the sort of random movements of molecules. They're always going to slowly spread out into a space or move from a place of higher concentration to a place of lower concentration. So this process of diffusion is always happening in all of your cells. And many multicellular organisms rely on the process of diffusion to move things around. The problem is the process of diffusion is way, way, way too slow to move stuff around and to get it to all the places that a large multicellular organism needs, so that it's each and all of its individual cells can get the nutrients it needs into it and the waste it needs out of it. And so on top of diffusion, multicellular organisms also rely on bulk flow to move things over much, much larger distances than diffusion alone, and at much faster rates than diffusion alone. Okay, which of the following is true? If you said C, mutation is the ultimate source of genetic variation in both eukaryotes and prokaryotes, that is true. So remember that whether a eukaryote or prokaryote, and you can move traits around either through horizontal transfer or vertical gene transfer, or if you're in the case of a eukaryote, you can do meiosis, you can have sex, and you can also sort of make new combinations of genes and alleles through those processes, or you can make new combinations of genes and alleles if you're a prokaryote through horizontal gene transfer. Ultimately, any new allele, any new gene has to ultimately come from a mutation that gives rise to a completely new trait that's never been seen anywhere in that species or anywhere in that population or anywhere on planet Earth before. So ultimately, any new trait, if you trace it far enough back, it has to come from a new mutation. So let's talk about why these other statements are false. Eukaryotes only have linear DNA in their genomes. This is false because although the majority of the DNA that makes up their genome that's in the nucleus is linear, they also have circular DNA in mitochondria and circular DNA, if they are photosynthetic, in chloroplasts. And so many, many eukaryotes, all eukaryotes and eukaryotic cells are also going to have circular DNA in their genomes because they have mitochondria, or if they're photosynthetic, they have chloroplasts that have their own circular DNA. And that counts as part of the eukaryotic genome. Prokaryotes cannot do vertical gene transfer. That is false. Vertical gene transfer is just from parent to offspring. Anytime a prokaryote does binary fission, they're going to pass an exact replica of their DNA to their offspring. So although they can do horizontal gene transfer, and that's the major source of genetic variation for prokaryotes, they can also transfer their genes vertically when they reproduce and produce daughter cells from parent cells through that process of binary fission. Horizontal gene transfer is the main source of genetic variation in eukaryotes. That is false. Eukaryotes on a sort of day-to-day basis are not capable of horizontal gene transfer. This is something that's unique to the prokaryotes, although we have observed it evolving 1 or 2 times in the sort of billion-year history of life. And that's when we had these processes of endosymbiosis, where a eukaryotic sort of precursor cell engulfed a prokaryotic organism that could do could do aerobic cellular respiration, or in the case of photosynthesis, when it engulfed an organism, a prokaryotic organism that could do photosynthesis, and that organism became a chloroplast. So horizontal gene transfer is unique to the prokaryotes. That's what I'm trying to say. Sex is the main source of genetic variation in prokaryotes. That is false. Only eukaryotes can do sex. Next question. Which of the following is true? Okay, if you said chloroplasts and mitochondria both have their own DNA, which can be used for DNA barcoding and maternity matching, that is true. So remember, chloroplasts and mitochondria evolved through endosymbiosis. So basically, chloroplasts and mitochondria used to be prokaryotic organisms, and therefore they have their own genomes. They have their own circular DNA. And you can use those genomes or the genetic sequences in there for DNA barcoding. In fact, that's what we use for DNA barcoding. Is for plants, we use part of the chloroplast circular genome. And to ID animals and invertebrates, we use part of the mitochondrial genome. And for maternity matching, remember we talked about that example where you could basically, mitochondrial DNA, that circular DNA that's in the mitochondria, is passed on from mothers to their daughters and sons through basically the fertilization of their eggs. And so if you look at the genetic sequence of particular regions of your mitochondrial DNA, that should match the mitochondrial DNA sequences of your mother and your grandmother. And we talked about this example of how we could, grandchildren who had been separated from their mothers and their mothers had passed away could be matched to their grandmothers with almost 100% certainty based on the DNA sequences of their mitochondria. So let's talk about why these other examples are false. Chloroplasts and mitochondria evolve through endosymbiosis, and so they're not examples of analogous traits that evolve through convergent evolution. So basically all eukaryotic cells have mitochondria. That's not because they evolved through convergent evolution. It's because they were passed on, and they are basically a homologous trait that's been passed on, and that trait arose through endosymbiosis, not convergent evolution. Chloroplasts and mitochondria both have linear DNA. That is false. It's circular DNA. And that circular DNA is what indicates that they evolved from prokaryotes through this process of endosymbiosis. So not linear DNA, circular DNA. Chloroplasts and mitochondria both have 3-cell membranes. No, they have 2-cell membranes, indicating they evolved from prokaryotes through this process of endosymbiosis. So remember, the idea here is chloroplasts and mitochondria at one point were independent prokaryotic organisms with their own cell membranes, and then they were engulfed by a parent cell, and when that parent cell sort of pinched out a membrane around them to engulf them through this endosymbiosis or this endocytosis process, so it's basically phagocytosis or eating another cell, it pinched off a little membrane around it, and so you'd predict there'd be 2 cell membranes around it, not 3, and so they have 2 cell membranes.